Hi everyone. This tutorial is going to explain how to use Trinity, which is a program involved in sequence assembly, specifically transcriptome assembly. So when we're studying the transcriptome, what we want to do is to sequence all the mRNAs in the cell, or all the RNAs in the cell. And so the way we do that is we extract all the RNAs and then we fragment them and sequence them in small fragments. So when we do this, what we end up with are files that look like this. So what we're looking at here is a series of reads. So each read is one independent sequence. So this is one, this is another, this is a third, this is a fourth. And so what we're looking at here is the top line you can see is just kind of a random identifier for the read. Not any information in there that I use, but it's kind of like the name of the read. And then we have the sequence of the read, and then an extra line. And this is actually the quality scores for the read. It tells us how good this sequence is. So let's focus in on this one. So you can see here this is about 100 nucleotides. In each one of these reads, as we go down, is about 100 nucleotides. So many of our mRNAs are several thousand nucleotides. So what this means is we're getting them all in very small chunks. So this might encode part of a protein, but it's only a very small part. The point of Trinity is to do an assembly of these sequences, so that basically we're going to take these small chunks and then assemble them into the full-length RNAs as much as is possible. So today I'm going to focus on actually how to do this on the command line, less about the theory behind sequence assembly. So let's see how this works. So I've just retrieved two files that are there to practice that are in the home directory. So you can see them if you just do an ls on the /home directory. And you'll see there are two files, one reads left and one reads right. So I copied those. They won't let you move them because you don't own those files. They're for everybody. So I copied them into my current directory. And so reads right and reads left, you can see they look very similar to each other. There's right and there's left. If we look at the top few lines of each one, that's the first few lines of reads right, and the first few lines of reads left. What we'll see is that they contain different sequences, but that they have matching identifiers. So this first line, you can see, has almost the exact same identifier as this first line. That's because these reads are a pair. Basically, these reads are two sequences that were generated by sequencing the two ends of a strand of a chunk of RNA. And so we're going to want to keep these together, and this gives us information about the positions of these sequences that the assembler is going to use. Okay, so we're going to then run Trinity on this. If we just type Trinity, you will see it spits a bunch of stuff on the screen. This is kind of a common way when you're trying to learn how to use a new program. It's a common way to try it. Often if you just type the program name, it'll give you some information. So in this case, there's all kinds of stuff that it tells us. It's giving us all these fairly intimidating bits of information about what we need to do. But then if we come down here, it gives us this helpful thing that says a typical Trinity command might be like this. And so let's go through what each one of these things is. So first we're going to type Trinity, and then we're going to have to tell it the sequence type. Well, the name of that file format that we were just looking at is FASTQ. So in this case, we want to put FQ for FASTQ, just like in the example. Max memory is just how much memory we're going to use on our computer, how much we're going to ask for the computer to allocate to this process that we're going to run. And for this case, we could do it with a very small amount. In general, you'd want to do something around the size of the data set that you're working with. So in this case, it's a very small data set, so we can just do 1 gigabase. And then we need to give it the left file, the left file, which we said was reads left.fastq, and the right file, which is reads.write.fastq, and then how many CPUs. Basically, a computer cluster, the one we've been using, is a bunch of different computers or different processing units that are hooked together. And in this case, we could use more than one of them if we want. So we could do, say, 4, something like that. It won't matter for it will matter for how fast it runs, but will not matter for the ultimate output. Okay, and so if we type this, it's actually going to run Trinity. And it's going to give all this stuff to the screen. This is basically, Trinity is running a series of different programs, and so it's telling us about the ones that it's running. So here it's in phase 1, so it's clustering the RNA-seq reads. It's looking for reads that are similar to each other. And then from there, it's building a catalog from reads. We can talk more about this, but this is basically a process where it's looking at the subsequences, the even smaller sequences that are within the read. And then it's constructing contigs, or extended sequences, from those short sequences, and so forth. So it's doing a whole bunch of different processes here. For today, I'm not going to dwell on these. If you're interested in the process of sequence assembly, you can find any number of tutorials online. We'll try to put one up on iLearn. But for our purposes right now, we just want to know sort of how do we run Trinity. And so, in fact, we have run Trinity. This would take a few minutes on this tiny little data set. So rather than our sitting here and waiting for the whole thing to go, I'm going to cancel the process, and we can go in and look at what was created. So by default, Trinity is going to create a bunch of different files within the Trinity out directory. I've made a copy of that from a run I ran previously, so that we wouldn't have to wait for it together. And so if we cd into that directory, we'll find a bunch of different things. And these are sort of the outputs of various of those stages that were going on one by one as we ran it. But what we want to focus in on is actually the ultimate output of this. The ultimate product is this trinity.fastday. And if we look at it, it gives us, we can see much longer sequences. Here's one that's 2,517 nucleotides. So now each one of these sequences represents a sequence assembly. What Trinity has concluded is an RNA. So here's one that's about 300 nucleotides. Here's one that's 2,517 nucleotides. So you'll notice that they have names, just like any FASTA file, and a bunch of information about the sequence assembly process. But you'll notice that these names are still kind of boring. They don't give us any information. That's because Trinity hasn't done any analysis of these sequences. It's simply done the assembly to tell us what sequences it sees. This is basically what sequences it's seeing in the reads that it's able to assemble. It hasn't done any functional analysis or seen whether there are coding sequences. That's up to us to do in subsequent analyses. Right, so if we go through, we find that there are many different sequences, and we can actually count those. So in this tiny little data set, it's given us 75 sequences, whereas in our initial data set, there were about 30,000 different sequences, or 60,000 different sequences. So it's taken this many different sequences and assembled them down to a small number, much smaller number. For your purposes, you'll probably be working with millions of reads and getting thousands of sequences out of Trinity, so that both your input and your output files will be much bigger. I would suggest to you that you run exactly this, run it on the practice set before you get going on your much bigger data sets. This will take some time to run, but hopefully that helps.